Bismillah ar-Rahman ar-Rahim. This is the first lecture in medical virology. Lectures in medical virology will include lectures on general virology, and then we will start systemic virology lecture. Systemic virology lecture will include DNA viruses and RNA viruses. Medical virology is the science that study viruses that cause a human infection. Viruses are the smallest infectious agents, having about 20 to 300 nanometer diameter. The viruses, also known as submicroscopic infectious entities. That is because they cannot be visualized with the usual light microscope, need to be seen by EM, by the electron microscope. So viruses, sometimes called submicroscopic infectious agents. Also the term filterable agent applied to viruses. This is because they can pass through 0.22 micron filter. This filter retains other microbes. For that reason, the virus is called the filterable agent, because they pass through this filter. The virus is an obligate intracellular parasite. They are chemically inert in the extracellular environment. They require living cells to support their replications. Generally speaking, the structure of the virus composed of only one type of a nucleic acid, either RNA or DNA, and this genome, the nucleic acid of the virus, is surrounded by a protein shell. This protein shell is called the capsid. Some viruses, in addition, they have a surrounding lipid layer called the envelope. Some definition in virology. Genome is the nucleic acid of the virus, as we said, either RNA or DNA. Capsid is the protein coat shell that encloses the nucleic acid genome of the virus. The function of the capsid, protection, and in case of naked viruses, they also serve for the attachment process in the way of the starting replication of the virus. The Capsomere, they are morphological unit of the capsid seen by the electron microscope, by the EM. Each capsomere is made up by one or more polypeptide chains. The envelope is the lipid containing membrane that surrounds some viruses. This envelope is derived from host cell membrane. Naked viruses are the viruses that without the envelope. The term Nucleocapsid is the protein plus nucleic acid. The protein and nucleic acid complex that representing the package form of the viral genome is called the Nucleocapsid. The Virion is the complete infective viral particle. In case of naked viruses, they represented by the Nucleocapsid, the Parvovirus, and this is the case also in coronaviruses. Whereas in the envelope viruses, the Virion is represented by the Nucleocapsid plus the envelope. This is the case in herpes viruses and in the orthomyxoviruses. General characteristic of viruses. First, the size. Viruses, they are the smallest infectious agent, as we said. 2. Location of replication, they are obligate intracellular parasite. They need viable host cell to start replication. They are totally inert outside the living host cell. 3. Genome contain only single type, either DNA or RNA. 4th, they contain specific structure. As we said, the nucleic acid, the genome of the virus, is enclosed by protein shell called the capsid, and some viruses also contain the envelope in the envelope viruses. 5th characteristic of viruses, the parasitic mechanism. The viruses are not capable of self-replication. They are parasitic at the genetic level of the cell. They compromise the machinery function of the infected host cell and shift them to produce viral particles, to produce a new complete virions. And lastly, the viruses do not affected by antibiotics. They need a special agent called the antiviral agents, include the antiviral drugs and the interferons. Now we talk about the chemical composition of the viruses in short. The chemical composition of the viruses include viral nucleic acid, viral capsid, viral envelope, and viral proteins. The chemical composition of the virus include viral genome, viral capsid, viral envelope in some viruses, and the other viral proteins. The viral nucleic acid, the genome of the virus contains single kind of a nucleic acid, either RNA or DNA. This genome code for genetic information necessary for the replication of the virus. The genome of the virus may be single strand, double strand, may be circular or linear, and the genome may be segmented or in one piece, non-segmented genome. The size of the viral genome, in case of DNA viruses, they range from 3.2 kilobase pair, this is the case in Hepadna viruses, to 375 kilobase pair in Pox viruses. Whereas the genome of the RNA viruses range from 7 kilobase in case of coronaviruses to 30 kilobase in coronavirus. The sequences and the composition of a nucleotide of the genome of each viral nucleic acid are distinctive. Now, the viral capsid. The viral capsid, they are symmetric protein shell surround the viral nucleic acid in the core of the virus. It's to protect the viral genome, and in case of naked viruses, it's important in the process of adsorption to the host cell receptor and penetration. Capsids are composed of varying numbers of capsomeres, the morphological units seen by the EM, each of which is made up by one or more polypeptide chains. The architecture of the virion is strongly influenced by the symmetry of the viral capsid. The architecture of the virion affected by the symmetry of the viral capsid. We have three types of viral capsid symmetry. Either one, helical symmetry, two, cubic or icosahedral symmetry, and three, the complex symmetry. the helical nucleocapsid symmetry of the viral capsid. Here, the nucleic acid of the virus has a spring-like or screw-like structure. This flexible configuration represents the helix by the nucleic acid of the virus, as shown in the picture. The nucleic acid, the genome of the virus, form a flexible screw-like structure, whereas the capsid protein is condensed on the helical nucleic acid. The light blue small pieces condensed on the nucleic acid on the screw-like structure is the capsomeres, the capsid protein. The helical symmetry of the viral capsid gives the virus a rod-like appearance. The helical symmetry of the viral capsid gives the virus a rod-like appearance. All the medical important human viruses do not exist as naked helical nucleocapsids. That is to say, in other words, all human viruses with helical symmetry are enveloped, and this includes orthomyxoviruses, paramyxoviruses, rhabdoviruses, filoviruses, arenaviruses, coronaviruses, and bunyaviruses. All of them are enveloped helical viruses. The second symmetry of the nucleocapsid is the icosahedral nucleocapsid symmetry. Here, the genome of the virus, surrounded by capsomeres, these capsomeres arranged in icosahedrons. The icosahedrons, a solid structure, a solid 3D structure with 12 vertices, 20 equilateral triangular faces, and contain 30 edges. The icosahedral viruses have a sphere-like appearance, as shown in the picture. The icosahedron form the sphere configuration of the viruses. Viruses with icosahedral symmetry include parvoviruses, papovaviruses, adenoviruses, pacornaviruses, calciviruses, and reoviruses. Most of viruses with icosahedral symmetry are naked. The exception is Hepadna viruses, herpes viruses, Toca viruses, and Flaviviruses. Now, the complex symmetry of the nucleocapsid. Here, the viruses cannot be categorized as having helical or icosahedral symmetry. So they said they have complex symmetry. Viruses are considered complex if they have complex structure, as in case of pox viruses, that is to say, cannot be categorized as helical or icosahedral, or they have the two types of nucleocapsid symmetry, the helical and the cubic symmetry at the same time. This is the case in retroviruses. So the two examples of complex symmetry of viruses is the pox viruses and the retroviruses. The pox viruses cannot be categorized as having having a cubic or icosahedral symmetry, whereas as cubic or helical symmetry, or in case of retroviruses, having the helical and the cubic symmetry at the same time. The picture represents the pox virus with complex nucleocapsid symmetry. Now the third chemical composition of the viruses is the viral envelope. A number of viruses contain lipid envelopes as part of their structure. The envelope of the virus derived from host cellular membrane, and it's acquired by the virus during the last stage of viral replication, as the progeny virus undergoes budding from the infected host cell. The chemical composition of the envelope consists of a lipid layer, a lipid bilayer derived from the phospholipid cellular membrane of the infected host cell. This lipid bilayer, decorated by viral coded glycoprotein, which are inserted in the lipid bilayer and projected to the outer surface. These glycoproteins on the surface of the virus serve two basic functions. One, they promote interaction with the nucleocapsid proteins, which is essential for the final stage of viral replication and assembly. Two, they mediate attachment to cellular receptors, which is essential for viral infectivity. The envelope viruses are easily dissolved by the organic solvents and detergents. The envelope viruses affected by the organic solvents and detergents because of the presence of the lipid bilayer on the surface of the virus, causing viral particle to lose its infectiveness. So the envelope viruses affected by organic solvents and detergents and lost its infectiveness due to the dissolving of the lipid bilayer on the surface of the virus by these chemical agents. Under structural proteins, these proteins essential for the formation of infective viral particles. Under structural proteins, we have some proteins are associated with the nucleic acid and responsible for proper packing of viral genome. This is the case of MP, the nucleocapsid protein. Other structural proteins, B, form the protective layer of the virus. Under this category, we have one, membrane or matrix protein, a relatively hydrophobic protein located on the inner side of the viral envelope of many envelope RNA viruses. This matrix protein is located beneath the external spike proteins of the viral envelope and beneath the plasma membrane of RNA virus producing cell. The viral nucleocapsid interact with the matrix protein in budding through the host cell membrane in the last stage of viral replication. The second example of the other structural protein from the protective layer of the virus and the outer surface of the virus is the spike proteins, the glycoprotein. They are viral coded envelope glycoproteins that project from the envelope surface. These proteins may have functions such as hemagglutination or neuraminidase activity or promote cell membrane fusion. to the other viral proteins is the non-structural protein, the enzymes. Examples of these non-structural protein enzymes: the viral polymerase A, viral polymerases. These enzymes are essential for nucleic acid replication. B, the proteases. These enzymes, the proteases, used by the virus to 1. Process large transcripts polyprotein into small functional proteins. 2. These proteases help the virus release nucleic acid from the nucleocapsid package of the core of the virus. C. Other type of enzymes is the endonucleases and ligases enzyme. They are specific enzymes used by some viruses in the process of replication. Some viruses enter inside the infected nucleus of the host cell and integrated with the genome of the host cell. In this case, the virus needs two enzymes, the endonuclease and the ligase, help the genome of the virus to be internalized inside the nucleus of the infected cell and be ligated with the genome of the infected host cell. Endonuclease and ligase enzymes. These pictures represent the different architecture of the virus. The upper two pictures is the helical symmetry and the two lower is the icosahedral symmetry. In the upper right picture represent the neck helical symmetry with the genome of the virus, with the nucleic acid of the virus forming a screw spring-like structure, and the protein subunits, the capsomeres, condensed on this nucleic acid. The upper left picture is the envelope helical symmetry virus with the envelope derived from the host cell membrane, and inside it the nucleoproteins and the capsid, and the capsid protein, the egg-like structure, is condensed around the genome, screw-like or spring-like genome of the virus. The lower two pictures, the lower right, the adenovirus with a neck non-envelope icosahedral symmetry, satellite-like structure. This is the adenovirus, necked icosahedral symmetry, and the lower left is the envelope icosahedral symmetry, the icosahedron, the solid 3D structure, the icosahedron surrounded by the two lipid bilayer, the envelope, and the envelope decorated with the viral glycoprotein. This is the case of herpes virus. That's to say, herpes virus having envelope icosahedral symmetry, whereas the adenovirus having a necked icosahedral symmetry. These two pictures represent the envelope helical symmetry in the right with the screw-like, spring-like nucleic acid or the genome of the virus, with the dot represented the capsid protein. This represents the nucleocapsid core of the virus, the nucleic acid, the genome of the virus, plus the protein package of the genome of the virus, the nucleocapsid, and this surrounded by the two lipid bilayer, the envelope, and this envelope is decorated with the glycoprotein, the envelope glycoprotein. Under the lipid bilayer of the envelope of the virus, we have the matrix protein, the matrix protein. So this was the envelope helical symmetry. In the left picture, we have the neck icosahedral symmetry in the core of the virus. Again, the genome of the virus plus the nucleoprotein. This represents the nucleocapsid core of the virus, and the icosahedron, the capsid protein, forming the icosahedral symmetry of the virus, and the viral glycoprotein being represent the viral attachment to protein to the receptor on the infected host cell. Now we will talk about the taxonomic classification of the viruses. The classification of the viruses can be based on 1. The nucleic acid type of the virus. Generally speaking, the virus, the family is the Viridae family, the subfamily is Virion, and the genus virus. So we can use the nucleic acid type as a basis for the classification of the viruses. In this case, we have 1. DNA viruses, and in DNA viruses we have, under DNA viruses we have Parvoviruses family. It's a single strand DNA virus. Example of the Parvoviruses family, we have Parvovirus B19. The second example of DNA viruses is Parvoviruses family. It's a double-stranded DNA. Example of these viruses is papilloma virus. Then we have the Adenoviruses family. Again, it's a double-stranded DNA viruses, and example of this virus we have the adenovirus. Herpes viridae family, double-stranded DNA viruses. Example of it is herpes simplex virus. Pox viridae family, double-stranded DNA. Example, the pox virus. Hepadna Toga family, it's a double-stranded DNA viruses, and example of these viruses is the hepatitis B virus. If we look to the DNA viruses, all of them are double-stranded DNA genome with the exception of Parvoviruses family. That is to say, only Parvovirus B19, the Parvoviruses family, contain a single strand DNA genome, whereas all other DNA viruses having a double-stranded DNA genome. And this is the picture of all the DNA viruses. Under RNA viruses, we have a long list of viruses, include Paramyxoviridae family. Example of these viruses, the respiratory syncytial virus. Orthomyxoviridae family, example of this virus, influenza viruses. Coronaviridae family, example of these viruses is the coronavirus. Pacornaviridae family, example of it, the rhinovirus. Rhabdoviridae family, rabies virus. Arenaviridae family, Lassa fever virus. Buniaviridae, the hantavirus. Calciviridae, the Norwalk virus. Filoviridae family, the Marburg virus. Flaviviridae family, the yellow fever virus. And Retroviridae family, the HIV virus. Under Togaviridae family, we have the rubella virus. And in Reoviridae family is the rotavirus. If we look to the RNA viruses, all of them are single-stranded RNA genome with the exception of Reoviridae family. That is to say, all the RNA viruses, they are single-stranded RNA genome with the exception of Reoviridae family, and the example of it, the rotavirus. And in the picture, representative of the different RNA viral family with the configuration, whether being helical or icosahedral, whether envelope or necked. The second way of classification of the viruses, we can use the physiochemical properties of the virion to classify the virus. Under this properties, we can use the molecular mass of the virion as a way of classification, the bionat density, الطفو of the virus, as the way of classification of the viruses, whether the virus is pH stable or not, the pH stability, the thermal stability, the susceptibility to physical and chemical agents, and the being affected by ether or detergents. This is the second way the physiochemical properties of the virion can be used to classify different viruses. The third base of classification of the virus, we can use the envelope, whether the virus is envelope or naked virion. This properties is combined with the symmetry. If we combine the envelope or not plus the symmetry, we have 5 different morphological forms of viruses, and these 5 groups, we have 1, the naked icosahedral viruses. Under it we have the parvoviruses, papovaviruses, adenoviruses, pacornaviruses, calci, and Reoviridae. So the Reoviridae, the rotavirus, they are naked icosahedral virus. The parvovirus, they are naked icosahedral virus. The second category in this classification, the envelope icosahedral, we have the herpes viruses, the Hepadna viruses, Toga viruses, and the Flaviviruses, the Flaviviridae family and the Flaviviruses. All of them are envelope icosahedral. Third category, the naked helical viruses. As we said, till now there is no medical important human virus exists as naked helical nucleocapsid symmetry. The fourth category is the envelope helical. All of the orthomyxovirus, paramyxovirus, rhabdovirus, phyllo, corona, arena, bunyaviridae are envelope helical viruses. So the coronaviridae family, the coronavirus, are envelope helical viruses. Fifth category is the complex, and as we said, the complex symmetry virus is the pox viruses in the Poxviridae family, and the Retroviridae family, the retroviruses. The fifth way of classification of the virus depending on the biological properties. These include the natural host range, the mode of transmission, vector relationship, pathogenicity, tissue tropism, the virus when enter inside the cell, where it go and start replication. This is called the tissue tropism of the virus, and the pathological change. Example of this classification under pathological change, we have a hepatitis virus, a large groups of viruses being categorized as hepatitis virus. That is to say, these viruses go to the liver and cause infection in the hepatocyte, causing hepatitis. Under the vector relationship, we have arboviruses. Arboviruses are a classification by the biological properties because they are arthropod-borne viruses. So all these viruses being said as arbovirus, arthropod-borne viruses, and so on. Now we have the current classification. The current classification, we have 5 basic subgroups of viral families. This derived by combining the nucleic acid type of the virus, the symmetry of the nucleocapsid, and whether the virus is enveloped or naked. These 5 groups, we have group 1, DNA viruses with icosahedral symmetry and naked. And under this group we have Parvoviridae family, Papoviridae family, and the Adenoviridae family. Group 2, DNA viruses with envelopes or complex code. We have the herpes viridae family, the Hepadna Viridae family, and the Pox Viridae family. Group 3, RNA viruses with icosahedral symmetry. We have the Pacornaviridae family, Calciviridae family, Reoviridae, Togaviridae, and Flaviviridae. Group 4, the RNA viruses with helical symmetry. We have Orthomyxoviridae, Paramyxoviridae, Rhabdoviridae, Filoviridae, Arenaviridae, Coronaviridae, and Bunyaviridae family. Group 5, RNA viruses with the mixed or complex symmetry, the Retroviridae family. This is the current classification. Lastly, we have a virus-like agents. Under virus-like agents, we have the prions and the viroids. The prions, they are infectious particles composed solely of a protein with no detectable nucleic acid. These prions highly resistant to heat, formaldehyde, and the UV and the ultraviolet. Disease caused by prions is called the transmissible spongiform encephalopathies. Example of transmissible spongiform encephalopathies, example of prions disease, the mad cow disease, and the Creutzfeldt Jakob disease. The other virus-like agents, the viroids, they are infectious particles composed of a nucleic acid without a protein code. They are RNA only. Example, the delta agent, the hepatitis D virus. And by this we end the first lecture in medical virology. And shukran jazilan. And thank you.